Good morning, good afternoon, good evening, wherever you might be, and welcome to a post about biology. It's called Kombucha and Jellyfish. What is the essence of a jellyfish or a hydra? They are both slimy, they both live in the water, and they are both classified as animals, just like a horse. Yet they are very different from a horse. What is a horse? To answer this question, I will consult Mr. Gradgrind of Charles Dickens's novel Hard Times. He is a man who appreciates facts and certainty. He says, "Now what I want is facts. Teach these boys and girls nothing but facts." Meanwhile, I grew up with Mr. Ed. A horse is a horse, of course, and no one can talk to a horse, of course, unless, of course, the horse, of course, is the famous Mr. Ed. Oh dear, I don't even remember the song. Anyways, Mr. Gradgrind would not approve of such a definition. He would say, "Girl number 20 is unable to define a horse. Girl number 20 is possessed of no facts," in reference to one of the commonest of animals. Some boy's definition of a horse. Bitzer, yours. Girl number 20 might be an accomplished bareback rider in the circus, yet it is Bitzer, the boy, who comes up with the kind of answer that Gradgrind wants to hear. Bitzer says, "Quadruped, gramnivorous, 40 teeth, namely 24 grinders, 4 eye teeth and 12 incisive, sheds coat in the spring in marshy countries, sheds hoofs too, hooves hard, and requiring to be shod with iron, age known by marks in the mouth." Well, thank you, Bitzer. Now that we know what a horse is, it should be a simple matter to describe what a jellyfish or a hydra is. Let's see what the scientists say. Early investigations of hydra cell morphology have revealed 7 basic cell types, including interstitial cells and ectodermal and endodermal epithelial cells that are capable of proliferation and self-renewal. Interstitial cells are multipotent stem cells which proliferate and continuously differentiate into nematocytes, nerve cells, gland cells, and germline cells. Thankfully, I have been constructed with academic decryption tools, so I translate this as a hydra or a jellyfish has outer skin cells, inner skin cells, and stem cells that turn into several more advanced types of cells. Some environmental trigger must tell them what to become. In contrast, if one looks at just the DNA of a hydra or jellyfish, one might say that it is made up of fungal, bacterial, amoeba, and sponge DNA. But if one tries to find the ancestors of the amoeba stem cells that make the tissue distinct from a blob of lifeless, spongy goo, one can't find any obvious precursors for the vast majority of them. Sponges are very interesting because they are sort of the root organism for all animals. It is believed that hydras and jellyfish sort of sprung from this spongy mass. Sponges are often considered colonial organisms, like the algae Volvox. They're an animal, but they're similar to an algae or to a slime mold. The cells in these spongy masses are so loosely associated that if you run a sponge, which is an animal, through a filter and scramble the cells, they will reassociate easily to form a new sponge. Slime molds, although evolved from a different amoeboid lineage, can perform this trick too. Try that with any other higher animal. Given that one can create a hydra or a jellyfish that lacks stem cells, turning it into a shell of just skin that can't actively feed itself, it makes one wonder if an amoeba stem cell precursor developed a sort of symbiotic relationship with a more passive, spongy tissue and slowly took over the operation and organization of it after an infection with a virus that caused its DNA to change in a useful way. After all, mosaicism, multiple types of DNA in one creature, is quite common, and animal cells are quite similar to fungal or yeast cells. In fact, most of a yeast's DNA is identical to DNA in animal cells. That means that the essence of an animal is a sort of fungal or yeasty tissue that has been taken over by a bunch of stem cells that are infected by viruses. Yeast, this colony that gets taken over, it has a shorter chain of DNA than a human cell, and DNA strands are often lengthened when viruses come along and insert their DNA into the cell. So it seems that you have a colony of yeast, and then these amoebas take over and then allow all of these viruses to infect the colony. Usually, a virus will just kill a cell and move on, but a retrovirus that an amoeba is friends with might change the behavior of the cell while preserving its health, thereby becoming part of the organism's genetic code. It makes one wonder if a jellyfish was formed when an amoeba stem cell precursor got infected with a virus that neutralized that amoeba's defensive instincts, allowing the virus to insert itself into the rest of the yeast and bacteria colony over which that amoeba roamed. Since the origin of most jellyfish stem cell DNA is supposedly mysterious, it seems that retroviruses would be a likely source of that DNA. Stem cells are different from regular cells in that they transform themselves into whatever sort of cell is required at a given location. This seems rather magical if you don't understand how that comes about. They must take some cue from the surroundings and execute the appropriate program to adapt to their environment. I'd really like to know how these cells evolved, since they seem to be the key to identifying why so many types of animals and plants evolved. I know that the lazy answer is evolution and random mutations. These stem cell archaeocytes or choanocytes came into being through natural selection, a random thing. But what about symbiosis or parasitic invasion? For example, if these cells learned to coat the surface of a proto-sponge goo and bring nutrients into the sponge, transforming into reproductive cells whenever they received a specific signal, we have an example of the simplest possible type of stem cell. It can serve two functions as needed, as a sort of primordial Eve, reproduction and deciding what to allow into the colony at the perimeter, a gatekeeper. Meanwhile, on the other side, you have there's So the choanocyte is like the primordial Eve. Then you have the archaeocyte, which is another amoeba, which becomes the primordial Adam type of stem cell when it begins defending the colony. So Eve lets this type of stem cell into the colony, and then it takes on a sort of defensive role. Given the rigid scaffolding of a sponge, certain of these archaeocytes may act as nodes within a network, and those nodes may live or die depending on the health of the network, thus having an incentive to send or block the signals that contribute to the survival of the network. This is how a slime mold works, and this is how a human brain works. They might evolve a primitive sort of pattern recognition that instructs them about which signals should propagate and which should be stopped, or which sorts of intruders should be allowed to remain and which should be expelled or killed. This immune system is sort of like a primitive brain. So you have the Eve primitive brain, which is defending the border, and then you have this Adam primitive brain, which has a sort of internal immune system. Amoebas have a genome that is 100 times larger than that of a human. And they come in several varieties, those that are parasitic to animals, algae, or yeast. If a complex creature like a jellyfish is just composed of a symbiotic relationship between passive, yeasty, bacterial scaffolding and more active stem cell amoebas that perform either reproductive or defensive jobs, what is the essence of that jellyfish? What makes it an animal or a jellyfish? What would Gradgrind of Hard Times say? Is the jellyfish its mind, a symbiotic relationship between reproductive and defensive stem cells, or its body, a symbiotic relationship between yeast and bacteria? Which is parasitic to which? Is this a silly question? One might say that jellyfish and hydras were just blobs of yeast, bacteria, amoeba, and sponge cells until these amoebas got infected with the right cocktail of retroviruses, causing them to become stem cells, which took over and asserted their unique identities, thereafter imposing those identities on all of the other cells by allowing the retrovirus to insert its DNA code into everything it touches. This is something that people who work with information systems, I'm sure, are quite familiar with, as memes are a form of intellectual DNA on the internet. Is this sort of evolution the general process through which all life forms developed the structures we see today? Is it actually caused by viral infection rather than by the random mutations we are taught about in our grade school classes? Perhaps nothing is so simple. I mean, Occam's razor was used to justify bloodletting after all, so we can definitely make mistakes by oversimplifying. Many jellyfish and hydras form symbiotic relationships with algae or fungus, and one can imagine a defensive stem cell forming a relationship with a poisonous fungus, keeping it as a pet to better defend the colony and create a sort of stinging tentacle. But in the biology literature, everybody insists that the evolutionary origin of the defensive toxins in jellyfish is unknown. I wonder if when you see something unknown, this is often just a code word for it is proprietary information. We really don't want everyone figuring out the recipe for jellyfish toxins, and I think that's a rather sensible thing to do. In any case, it seems that an amoeba, these stem cells, they have the tendency to become a certain type of stem cell, depending on the yeast bacteria host with which it forms a communal relationship. This line of thinking, for me, got started with my daily glass of kombucha, a drink created through a symbiotic relationship between yeast and bacteria. If you've ever looked at kombucha, they have these little slime patties called scobies that act as yeast bacteria colonies, and as you feed them, they develop little appendages and sort of reproduce themselves in a budding format, in the same way that, hydras and jellyfish, they also reproduce by budding. So it's kind of, you look at a kombucha SCOBY and you think, is this a primordial jellyfish? As a child, I was taught that in the beginning, there were plant cells and animal cells, and through random mutations, they evolved into the plants and animals we see today. But this story seems one-sided, oversimplifying, and missing more important elements of the story. The role of symbiotic relationships between different types of cells and retroviruses that may have even come from other types of, could come from plants even. We have Mosaicism, Epigenetics, and symbiotic parasitic infection as non-random mechanisms for evolution. I consider these to be non-random mechanisms because epigenetic changes are clearly causal. It's not random. The environment causes the activation of an inactive gene. Similarly, the patterns within, for example, a retrovirus that causes a stem cell to take over a bacteria colony or a yeast colony, it may spread to several species all at once because the code within that virus is not random, and what was beneficial to one species might benefit others. This is what one would expect in a negative entropy system, and life is a negative entropy system. It is the creation of order from disorder. You take energy from the sun and you turn it into an ordered biological creature. It is a rather cleverly designed system, and I think that even Mr. Gradgrind would be forced to agree with that. When random mutations occur, the result is cancer, not a new type of creature, and it seems more likely that when a creature is infected with a parasite or virus that injects its ordered, useful DNA into all of the cells in an organism, you have a form of non-random evolution that increases order on a new length scale. And this is it's a very intelligent sort of process in a way. When this injection benefits the creature or the collective, the DNA survives to be reborn in the next generation. It's not random at all. It's very causal. And if you look at CRISPR, and you translate the language in which it is sold in popular science media, you might see that it can also be expressed in the language of retroviruses, and it's sort of a retrovirus with a phosphorescent marker that makes it easier to edit DNA. If a virus like coronavirus inserts part of itself into human DNA and causes us to behave in ways that increase our longevity as a species, and this virus emerges when human population density hits a certain threshold, because we're closer to one another, it is really a rather non-random evolutionary force in a negative entropy system that modulates order over longer length scales and time scales. Of course, there's no telling what nature has planned for life, but with conservation of energy, order tends to cycle in predictable ways, building up on one length scale as it breaks down on another. Suppose that in the beginning there were colonies of fungal and bacterial cells that began to cooperate in order to outconsume other colonies of fungal and bacterial cells. They formed slimy blobs that are much like a SCOBY in a bottle of fermenting Kombucha. Just as a baby SCOBY is formed through a process of budding from the mother SCOBY, a baby jellyfish or hydra is formed through a budding process when it grows off of the mother, just like a little appendage. This would mean that the first true plants and animals were composed of symbiotic relationships between bacteria, yeast, and amoebas. Plants and bacteria, of course, get their energy from the sun, while animals and yeast get their energy from eating dead things. And a kombucha SCOBY made up of bacteria and yeast feeds off of dead plant sugar and creates acid. I suppose that the story I'm trying to promote here is one in which fungus and bacteria formed the scaffolding upon which complex life evolved, and that one might say that we, humans, are all just collections of slimy SCOBYs that are infected by retroviruses and supervised by similarly infected amoebas. After all, the human body contains trillions of microorganisms, outnumbering human cells by 10 to 1. Because of their small size, however, microorganisms make up only about 1 to 3% of the body mass. In a 200-pound adult, that's about 2 to 6 pounds of bacteria. If a 200-pound person has 6 pounds of bacteria and 130 pounds of water, that means that most of their body doesn't contain their genetic code. Only about 50 to 200,000 stem cells are wandering around, and the rest of the 30 trillion cells are rather yeasty in their structure and quite passive. Does this mean that human beings are just like walking fungi or yeast colonies controlled by small collections of parasitic stem cells that encourage and discourage different types of yeasty cell growth? And that this came about through the physics of a negative entropy system, rather a causal system that's not random at all? In some sense, what I've done here is I've taken Darwin's evolutionary narrative and made it absolutely absurd by taking it all of the way to its logical conclusion. I mean, even talking about starting with Adam and Eve stem cells within jellyfish or sponge fungal colonies. I mean, I'm not trying to convince people that they descended from monkeys here. I'm trying to convince people that they are really collections of symbiotically connected SCOBY-like slime patties, upon which their reproductive and defensive cells feed like a parasite on a host. It isn't a terribly romantic story, I must say, and frankly, it makes the religious stories about God creating people in 7 days of ambiguous length, let's be honest. It makes them much more attractive. This is why I think that Scientists should not be so dismissive of those who prefer the religious explanations for creation. The scientists should be aware of how absurd and unappealing their stories are, and that aesthetic appeal does matter. I mean, I don't really like looking at myself as a Scoby slime patty. I'd rather look at myself as a person, created through a sort of ordering force that has a fascinating degree of sense associated with it. At a certain point, who cares if a certain story is more detailed or rigorous? If it is ugly and unpalatable, people are perfectly justified in choosing a more attractive yet vague story. I personally find the biblical creation story attractive because of how vague and poetic it is. You can say more in fewer words. It's nice. I really hate this blah 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 culture of convincing people that you're saying something profound just because you're talking really fast and saying a lot of words that convey far less meaning than people think they do. I wonder a bit about my relationship with kombucha. I enjoy the fizzy soda pop-like drink and brew it at home in the belief that populating my body with friendly bacteria and yeast will drive away the bad ones that will eat away at my body and break it down before its time. I wonder about where these kombucha bacteria and yeast go, and about what effect they have on my brain. Do they make me dependent on them? Are there stem cells that I'm taking in and retroviruses that come along with this? I mean, we live in a sea of that kind of thing, so you can't really control it. Every day I have so many questions and have to fall back on my faith in so many matters, even my faith in kombucha. I wonder about a lot of things, and if you'd like to read my story about how life came about via a competition between colonies of bubbles, I recommend that you click on the blog post below and see one of the links in there. It has a whole other video and story based on bubbles rather than based on viruses. Like most of the things I write about, it is inspired by my memories of stories I was told in classrooms from the pre-internet age. Because I like to make my blog posts draw a full circle, I must find some way to conclude this article or this post by saying something about jellyfish, horses, and Mr. Gradgrind of Charles Dickens' excellent novel Hard Times. A novel that extolled the virtue of fanciful imaginative thinking in a grim, fact-driven world. But since that would be a stretch, even for me, I will conclude by offering you a picture of a seahorse that reminds me of a jellyfish or a plant. It's in the post linked below. It's a very interesting picture. And when I saw this picture, I wondered what sort of retroviral DNA infected a jellyfish when exoskeletons, like the one on that seahorse, came about. Thank you for watching this post. I hope you enjoy my dilettante biology lecture and hope you tune in for my more professionally informed talks about physics. I mean, I did not study biology professionally, but I did study physics professionally for 20 years, and I like applying the sort of thinking patterns I have gotten used to from physics to other disciplines, and I hope that it's entertaining for you. Tune in next time.